My guest today is Professor Trey Ideker, who is Professor of Medicine, Bioengineering, and Computer Science at the University of California, San Diego. He is also the director of the Bridge to AI Functional Genomics Data Generation Program and co-director of the Cancer Cell Map Initiative. And I understand, Trey, that DARPA has a new initiative that you're part of as well, which is an exciting predictive oncology initiative. So we'll talk about this toward the back end of it, but I want to start with one of your papers. Desmosome mutations impact the tumor microenvironment to promote melanoma proliferation. So you say here desmosomes are transmembrane protein complexes that contribute to cell-to-cell adhesion in epithelia and other tissues. Here we report the discovery of frequent genetic alterations in the desmosome in human cancers, with the strongest signal seen in cutaneous melanoma, where desmosomes are mutated in more than 70% of the cases. Yeah, I mean, that is an amazing statistic. Without knowing much about this, this should have both diagnostic as well as potential early intervention possibilities. If so, let me ask you this: can we do a blood test to determine this?
You can do a biopsy of the melanoma tumor. So since that's your skin, biopsying melanoma is routine. You know, it's accessible tissue. It's routine. And what you would do from the biopsy is you would sequence the DNA of the tumor, as is now currently done in many cancer clinics, and look at the desmosome to see whether the genes encoding the desmosome have mutations. As you just pointed out, the main exciting discovery of this paper is that more than 70% of melanoma tumors have compromises to the desmosome complex. And it had been utterly missed in all of the previous analyses because the mutations are not in one gene, but they're spread out over about 15 genes that make up this system in the cell, or it's actually, it's important for how cells attach to one another in skin. So it's happening right at the cell membranes to connect cells together to form a dense layer of keratinocytes that forms your skin tissue. This had never been seen before because the mutation burden is really spread all over the 15 genes. It's not just in one.
That's really interesting. So I think public understands sort of gene mutation and related issues in many of the diseases, but this is a little bit different, isn't it? I mean, so desmosomes, you say, is a protein complex, and rather than mutation, you say here genetic alteration in the desmosome in human cancers. So what are the implications of that?
So first of all, genetic alteration is just a more general word that encompasses mutation and also so-called copy number alterations. So let's just talk about that. Actually, many of these events are nucleotide mutations, changing from an A into a C, G, or T, just like you expected. But some of the events are larger, what are called structural variations that you see in the tumor genome. Entire arms of chromosomes can be lost or gained, for instance, or sections thereof. And when that happens, all the genes, you can imagine the genes are like sitting on the chromosome like a number line, 1, 2, 3, 4, and so on. If I take a big chunk of DNA that might have 100 genes on it, and all of a sudden I lose it, which happens often in the early stages of cancer, because one of the hallmarks of cancer is you lose so-called genome stability and your chromosomes go wild, basically. And so when this happens, you might lose 100 genes at a time off of one chromosome, or duplicate. It can also happen. You duplicate all of those genes by retaining an extra copy of that segment. So anyway, these are all generally called genetic alterations. And just as as lots of previous studies have found, we find that both point mutations and larger rearrangements can affect this desmosome complex. Now, in terms of people are used to thinking about genes, it's not just the lay public that's used to thinking about genes, it's scientists that are used to thinking about genes. And so if you think about cancer as a gene level disease, there have now been hundreds of genes that have been in which alterations, mutations or copy number changes have been associated with cancer, and numerous of those with melanoma. But that's essentially treating each gene as an independent part of the system, which it turns out misses a lot of important changes that happen. And the reason it does is because each gene you can think about as it makes one protein or one type of protein. And what do those proteins do inside of the tumor cell? They're different subunits of machines that make up a tumor cell, much like you think about the nuts and bolts of your car. And mutation or removal of any one of those nuts and bolts may be part of the explanation, but it's not really what's happening to cause cancer. What's happening to cause cancer, the infamous analogy with cars is cancer has to have some way of destroying your braking system. At the same time, it has to have another way of mashing down on the acceleration or accelerating the car. Because what is cancer? It's nothing if not a car out of control, screeching down the freeway at 140 miles an hour until one day it crashes. You know, it's cell proliferation gone horribly wrong, right? So it's uncontrolled speed, essentially. And so how many ways are there of taking out the braking system or causing the car to accelerate without control? Well, I can literally put a brick on the accelerator, or I can literally take a hacksaw and hack off the brake, okay? So call that one gene, right? But downstream of the brake pedal, as you know, there is a whole braking system. So there is the hydraulic system through which brake fluid is pumped. Maybe what I've done instead is I've drilled a hole in that hydraulic tubing, or I've somehow otherwise depleted the braking fluid. Or alternatively, maybe I'm downstream of that and I've removed a brake rotor at the wheel. And so these are all different ways I could destroy a braking system so that a car can screech out of control down the freeway. In that exact same way, there's many ways to disrupt what are called a hallmark pathway of cancer. And so the attention, while it's been on these gene subunits, it really should be on the system. How is it that this system is taken out? So in this case, the system is how cells attach to one another through this desmosome junction, and it's got 15 subunits that make up that braking system, if you like. So there's 15 roads to Rome when it comes to how you damage this system, and in taking a machine level or a systems level view of cancer, we were able to see findings like this. And in fact, this is one of several publications we've had recently where you find a systems level effect that you didn't see at the gene level.
Yeah. So you say here in primary but not metastatic melanoma biopsies, the burden of coding mutations in desmosome genes is associated with a strong reduction in desmosome gene expression. So I'm thinking practically and economically, a biopsy is much more difficult. Is there any other way we can sort of early identify the issue?
It could be. You might be aware, Gill, there's a series of emerging tests known as multi-cancer early detection, MCED, sometimes pronounced MCED. There are a variety of companies that are out there on the market right now for different kinds of early cancer detection tests. I won't advertise any of them here, but nonetheless, a simple web search will reveal a number of ways of getting the screening done as a patient or as a consumer. The question with this particular finding would be, should we represent the desmosome in in those early screens for melanoma? It's not it's our paper's early. It's the initial discovery. What you're doing is anticipating what are some downstream implications and screening, given that we found that the vast majority of melanomas need to have, it looks like, if I can anthropomorphize cancer, it sort of needs to have this mutation occur. And in retrospect, it makes perfect sense why you would need to weaken cell-cell junctions. It turns out the real big surprise of this paper after the initial finding was that the mutations look like they're not in the tumor itself. The tumor is in what's called a melanocyte. When you get skin cancer, it's in the type of cell called a melanocyte, and that's why it's called melanoma. But melanocytes exist surrounded by what's called the stroma around the tumor, and they're consisting primarily of another cell type called the keratinocyte. We've of course heard of keratin, which really forms these keratin fibers, form our skin. And so if you think about it, the melanoma cell, as it forms the tumor, has to grow and press out against these surrounding cells, and you need room for it. It needs to make room. And especially once it metastasizes, which of course means that at that point in metastasis, the tumor itself is broken completely free and now is free to travel throughout the body. But to do that, it's going to have to, again, break free of all of these surrounding keratinocytes or make its way through them. So it makes perfect sense why you would need to weaken this cell-cell junction in the surrounding tissue.
So one of the important things here is it's a system effect, as you talked about, the braking system, the acceleration of the vehicle. It's a system effect, and it's very difficult to pin it down to a singular mutation, possibly. Is this true with, so we have this breast cancer, ovarian cancer, BRCA mutation. Is that something different?
It's going to happen in all of cancers to a first approximation. There are a few cancers which are so-called monogenic diseases, meaning they're really attributable to a single genetic defect or aberration. But the vast majority of cancers, and almost all the common ones, are systems level effects. This is not a new idea, as you might have alluded to. One of the most transformative scientific articles ever published in the cancer field is an article by Hanahan and Weinberg called "The Hallmarks of Cancer" that came out more than 20 years ago now, and already, I mean, so you think about this article comes out at the beginning of this millennium and really throws down the gauntlet that we should be thinking about cancer not in terms of genes or individual mutations, but as what they called the hallmarks, which is exactly like the braking system and the acceleration system, and the different systems of a cell that need to be disrupted in order to permit this horrible disease. So really, our paper is the next installment, if you like, in the Hallmarks of Cancer story. Yeah. And some of the other work from my lab that we'll talk about perhaps later is also really not the first by any means to talk about disease in terms of these hallmark systems. It's really, how do we implement that idea practically, and what are those systems?
Yeah, so I was in a large pharmaceutical company, Trey, in the late 90s and early 2000s. As you know, there was a lot of excitement about the Human Genome Project, and half the crowd walking around saying all diseases are cured in like a couple of years because we know exactly where the mutation is and exactly how to treat it. 25 years later, I don't think we have cured anything. So it is not reductionist in that way, right? I mean, our tendency from an engineering perspective is to reduce it to simple equations, but biology is not really amenable to simplification that way. That's the way that I'm thinking about it. Yeah.
So first of all, I think it's a bit pessimistic to say we haven't cured anything or learned. Certainly, we've learned an enormous amount from the human genome, and the human genome knowledge of that and the analysis of the genome has led to many cancer treatments and continues to do so, as well as for other major diseases. So I think the issue isn't so much that it hasn't done anything for us, but I think what you're expressing is that it's clear that we've just scratched the surface.
Yeah.
Of what is possible, and that there's going to be an enormous amount of work and perhaps conceptual breakthroughs that need to happen for the information in our DNA to really be decoded, as was promised more than 20 years ago. And so, that is exactly what, understanding of these hallmarks is not the only, perhaps, domino that needs to fall to really break open the human genome and reveal and cure all diseases. But we certainly think it's a significant part of the puzzle that has been overlooked or and still needs to be filled in. Right.
I mean, it's clearly data. I mean, data is always helpful. But I think what you're showcasing here is that the system effects, they tend to be very nonlinear. They tend to be very multimodal effects. And so it is not really a linear engineering question like we thought in late 90s. And so this Desmosome study here is sort of reinforcing that idea, right? It's so at some point people said, okay, you got the human genome right data, but it is really about proteomics. I mean, you still have to figure out what proteins are in there and what they're going to do, how they're created, and all that stuff.
Well, let's look at this from again an engineering perspective, though. So we have a system called the human body, and the idea of genomics is we'll look at changes in A, C, G, and T letters. And so as a physics problem, what are the scales of those letters? Well, an A, C, G, or T is, let's say, around a nanometer in size, maybe sub-nanometer in size. And you're trying to connect those changes to events that happen at the level of the patient. So this patient has cancer or not, or this patient has diabetes or not, or heart disease risk. These are events that are for objects at the meter scale, the scale of a person,
Yeah.
What we're now talking about is filling in the gaps between the nanometer scale and the meter scale. Did we really think, if you go back to the prospect of genomics 20 years ago, did we really imagine that we were going to be able to directly connect these two scales without any intervening knowledge? 9 orders of magnitude of gap in physics were going to be left out.
Some people did.
Yes, exactly. Then you're also alluding to another approach that perhaps you were working on. Certainly, I was part of this movement maybe in the 90s, which was, okay, we get that there's information, there's physics in here between the nanometer and meter scale. Let's write it all down with ordinary differential equations in a series of in a very large model, right?
Yes.
And this also has failed. And so that was an approach that was tried early and maybe certainly has some limited use. Let's not completely throw that under the bus. But why generally does it not scale? Well, it requires first of all, it requires us to know the forms of all of the equations that the biophysics of our bodies uses. Even if we did know what all those equations were and could write them down, those equations have millions of parameters that we would need to estimate somehow. and so on and so forth. And so here is where machine learning and artificial intelligence in its modern incarnation has been, perhaps I would argue, a more suitable mathematical framework. Because
to reduce the complexity some ways.
Exactly. And also to exactly, to reduce the complexity, but also to allow estimation of hidden variables that we have no way of measuring. Right. So yeah.
Sorry to interrupt. So an alternative hypothesis, Trey, would be that God has a sense of humor. She sort of guides you down a path, and then when you get closer to what you think is the truth, she drops you. This has happened to humanity, for the past 10,000 years, and continues to happen. But reducing complexity is possibly a way to get around it. You think?
I think it's got to involve reduction in complexity almost inherently to the problem. Let's go back to the multi-scale nature of converting the genome into disease. So at the nanometer scale, again, we have many different ways you can mutate a human genome. Let's look at that complexity. There's 3 billion base pairs, as we've famously been taught. Now, it turns out not all of those can be mutated without killing the organism, but there are nonetheless, let's say, of those 3 billion letters, there's certainly millions that can be changed to precipitate cancer development, or certainly to participate in cancer evolution. So, and as you further noted, those million ways don't happen independently or don't act independently from one another. They can combine in nonlinear, unexpected ways. Okay, so that's a very complex situation at the genome scale, nanometer scale. Let's look at the meter scale. Patient cancer, and treatment is either treated successfully or unsuccessfully. Pretty simple. And going back the other way, cancer has been described as being many diseases, all of which have the same label, cancer. And that's simply another way of stating this. It's highly complex at the molecular level. Situation is unfortunately quite simple at the meter scale.
Because the end outcome is binary.
Exactly. So clearly, high complexity must be projected into low complexity across the scales of cell and tissue biology. And that, in another way, is exactly what we've been talking about since you brought up this Desmosome paper, right? The Desmosome is one of maybe a few hundred objects that cancer will change in order to precipitate its evolution, cause itself, right? But we've already vastly reduced the information from millions of letters of DNA now convergent upon just a few hundred systems at the inside of the cell.
Right. So I didn't quite understand this, Trey. So is the cancer changing the desmosome, or the changes in the desmosome precipitate the cancer, or both?
Well, let's be good scientists and never anthropomorphize. I'm already on this call guilty of anthropomorphizing cancer several times. So the cancer doesn't want anything or do anything or need anything. So I think it's the other phrase you said, that these changes will promote cancer. Now the question still remains: is it are these early changes or late stages in the development of cancer? And this question is thus far unanswered. We don't know. We have, in fact, if you read the discussion of our paper, we thought a lot about this in interpreting our results. We can certainly see an early event hypothesis where, as you live your life cancer-free, your skin is exposed to UV rays when you go out in the sunlight too long. But of course, as careful as one is, it's impossible to avoid UV damage on one's skin. And maybe what's happening is over time, from almost birth, we're beginning to get genetic alterations in the desmosome, such that one day, in a particularly weak part of your skin, a melanocyte blooms into cancer. That was a significant hypothesis that we considered in our closing of our paper.
So I want to go to another paper, Trey, another very interesting paper. So multimodal cell maps as a foundation for structural and functional genomics. You say here human cells consist of a complex hierarchy of components, many of which remain unexplored. Here we construct a global map of human subcellular architecture through joint measurement of biophysical interactions and immunofluorescence images for over 5,100 proteins in the osteosarcoma cells. So let me try to at least peripherally understand this. Right? So you say human cells consist of a complex hierarchy of components. So from a naive perspective, the cell is a pretty complex machine, isn't it? So there are a lot of things in there. And I understand that how they interact with each other. I mean, it's like a Swiss watch of some sort. There are a lot of components, turning and, getting energy or putting energy out or whatever. And they all have to work so perfectly for the cell to work. So it's a miracle that we survive, isn't it?
The Swiss watch is a great analogy. Arguably, the cell is far more complex than the most complex watch. Arguably, it's also more error tolerant. One of the only ways this thing does function is it has quite a bit of ability to respond to damage and to errors of various kinds. And how to map the structure is still a frontier of science. Unlike the genome, which was fully sequenced, or at least the first full sequence paper was published more than 20 years ago. Turned out that wasn't the whole story, but was updated several times since. Nonetheless, we've been sitting on that sequence for 20 years. We are still far from revealing a complete map of a human cell. If you think about it, the DNA itself is already part of that map. The DNA itself is the thread of genetic information in the nucleus. So it's part of that map. But now, what else is in the nucleus? And as you've noted, it's not just DNA or RNA. It's also protein. It's also so-called small molecules like metabolites and lipids. And they all, back in the 50s and 60s, the model of a cell was basically it's a big balloon or a bag full of enzymes, and they're just kind of swimming around in free diffusion. It turns out that, as you said, just as God leads us to one particular conclusion, then it's overturned in the next scientific revolution, and so cell biology has since moved on quite significantly from that idea of just a few bags of fluid inside of each other. In fact, it's much more like a Jetsons-like city. It's some Manhattan or maybe your Swiss watch of stuff of all manner of stuff flying around in a fairly organized way, and so cargoes are constantly trafficked in a very directed way from one part of the cell to another to build these machines, and as a consequence of their function. So really one should think of like a city or a watch, How to map this is what this paper is about, and it's not like it's the next installment in this question. It's not even yet the final map.
Yeah, it's very much like a city in the sense that there's sort of uncertainty in there too. As you say, it has self-correcting mechanisms built into it. When there's a traffic jam, it can do something to correct the traffic jam and so on and so forth. So you say here self-supervised multimodal data integration resolves 275 molecular assemblies spanning the range of 10 to 8 or 10 to 5 meters, which we validate systematically using whole cell size exclusion chromatography. Maybe I got the units wrong here. So 275 molecular assemblies spanning the range. So talk a little bit about that. Yeah.
Yeah. So the way that we mapped a human cell, this was a proof of concept for how one can map relatively quickly. And by relatively quickly, I mean with a few years of work. One could imagine making this much faster, in fact, but in a few years of work, how can we best elucidate all of the components that are inside of that cell machine?
Yeah.
And the way that we did it was we applied two technologies in tandem. That's the multimodal part of, and the word multimodal, self-supervised, is in reference to a exciting machine learning approach that a lot of people are using, a lot of companies and labs are using. Sometimes these are called foundation models. You'll see this in the literature also. So in that sense, this is a foundation model of a cell.
Like the large language models.
Models are many of them are foundation models, self-supervised as well. Exactly. So here we used two different data streams. One is, you can think about the leading edge of biochemistry approaches, and the other is the leading edge of imaging or microscopy approaches. So what do microscopy experimentalists do? They look at cells under glass, in 2 dimensions. Actually, they get a third dimension where they can scan up, and we've applied this to about 5,200 proteins in a model cell type, as we describe in the paper. This happens to be a tumor cell, so it actually feeds right back into this discussion and our primary motivation of understanding how to interpret and read the tumor genome so we can solve cancer, right? So the fact we chose a tumor cell as our model was not by accident here, but in the tumor cell, we were able to map 5,200 proteins with microscopy in terms of lighting them up in cells and seeing where they go. And then the other main technology is the biochemical approach. Well, what does biochemistry do? Well, biochemists don't look at things under microscopes. They, to a first order, grind things up in blenders, pour them over so-called columns, and look at what sticks. And so if I grind up the cell population, extract the proteins from it, and I pour it through essentially think about it, it's named a column, like the sort of Greek or Roman column, but it's a hollow, think about it like a hollow tube. And in the tube, stuck, sort of packed in there, is a particular antibody against a protein of interest. And basically what you want to do is you want to isolate one protein as well as anything it might be sticking to, and identify those interacting protein partners. And that kind of tells you in the Swiss watch, okay, this bolt goes in this hole and attaches to, allowing you to attach this part. And so you get these sort of these binary or pairwise interaction diagrams of what sticks to what out of that biochemical data. Whereas the imaging is much is very complementary. It tells you what proteins are in similar regions of the cell, but they're both kinds of information about a protein's position.
Yeah, I think this is the direction humanity is heading. Now we have multiple techniques, visual, large language models, machine learning, and so on. So combining them in an effective way to reach something more interesting, I think is where the value is going to be. You say here it decodes pediatric cancer genomes.
Yes.
identifying 21 recurrently mutated assemblies and implicating 102 validated new cancer proteins. So that that seems potentially diagnostic as well, right?
And it was exactly inspired by the Desmosome paper that you mentioned already. The Desmosome was one such component. And given now that we've created this map of the majority of proteins in a cancer cell and where they reside and what machines they form, how many other machines like the Desmosome could we find where you get this convergent pressure of mutations from all sorts of different people's tumors?
Yeah, it's mind-boggling. I don't know why humans, well, I know the answer, but we went down this path. The further we go, the more complex it looks like. And it's really difficult, I think, from a systemic perspective to tease it out, right? I mean, we find more and more interesting things in the cell, in the human body, and on all biological systems.
Absolutely. In fact, it was
Yeah.
a surprise to me. About half of the 275 objects that you mentioned, we were able to define these. We can resolve them clearly in the data. Now the question is, are they known to humanity already? If I open my Cell Biology 101 textbook, do I see them there, or do I not see them there? And it turns out it's almost half. Or the division there is about 50-50. So about 50% of those map to well-known objects of cell biology. And again, spanning scales. This is, so we recover the nucleus. The largest objects we recover are organelles. We recover the nucleus. We recover the mitochondrion. We recover the endoplasmic reticulum, and so on. But then within each of those, you go down to the next scale and look at what objects you see. Well, within the nucleus, you have, for instance, a well-known part called the nucleolus. And then you have chromatin, and chromatin has compartments, and then within that smaller complexes, and you can recover all of that knowledge to a first approximation. But now let's look at the other half of objects in this map we created. They simply did not correspond to any known components that were previously well beyond Cell Bio 101, previously published in any literature. We've worked a few of these up for the paper, but by and large, most of them are still unknown. They're in the map. We gave them systematic coordinates. We weren't comfortable naming them much more deeply than that.
Yeah, I mean, as an educator, Trey, this is not your research, but I want to ask you sort of a conceptual question. So when information changes every day, every hour, every minute, it's sort of an interesting thing for an educator, right? So you go to the course that you're giving on Monday morning, you go in and you talk about what we know. By Friday, possibly the information has changed. When I was going through engineering school, nothing ever changed. We use the same textbooks. Even physics never changed. So as an educator, this is sort of a bit of a challenge, isn't it? I mean, your information is changing dynamically. So how do you teach kids about them?
Well, I think there are certainly truths about biological truths that are well vetted that one still teaches. But your point, for instance, the cell still has a mitochondrion, and still, which still carries out oxidative phosphorylation in terms of the main metabolic engine of our cells, and this is not changed. But it's simply that the situation is more complex than had been revealed, and I think you're commenting about a couple of things that science and scientific progress is accelerating over time. It's moving faster every year that passes. for a variety of reasons, and this gets into the singularity and all of that.
Yes.
So how to so teaching, I think I'm not so worried about, because you still teach. There's still plenty to teach that is knowledge today, and is going to still be knowledge in 20 years. It's
That's
highly relevant.
foundational. The
Foundational.
foundations don't change. Yeah.
Exactly. But well, and maybe the guideline as an educator is make sure that what you're teaching has that property. It's foundational, and maybe spend less time on well, unfortunately or fortunately, that would mean, if you followed my advice, spending less time on late breaking results.
Yeah.
Now, of course, one can imagine the virtue of teaching some late breaking results as well. And for instance, at the graduate student level and above, you spend a lot of your education time in journal clubs, where you're reviewing late breaking papers and discussing those papers as a manner of keeping current. And even that's getting difficult. I would almost say that's keeping current in science is really what I worry about. It's not so much the challenge of what you teach, it's how in the world are scientists going to continue pushing forward? Is the scientific process going to break because there's simply no way for a human to track all of the information that came out yesterday about your discipline?
Or even practitioners. I often wondered about this. Doctors, for instance, they're practicing, they're seeing patients every day. How do they keep track of all the information that's coming out, which sometimes is contradictory, and what they learned in medical school oftentimes is obsolete. It's a really a tough environment from that perspective when you have this steep slope in knowledge and information.
Well, and so this is a great segue to one of the centers I'm involved with that you had mentioned at your introduction, which is trying to solve this problem in the cancer clinic, in terms of how do we take up-to-date knowledge about cancer drug responses and cancer, the architecture of cancer as a disease, and use that up-to-date knowledge for treating cancer patients today that are in need. And that question was the answer. One answer is you use AI, right? That's one answer. It's not the only answer, and it's not necessarily an answer that's not fraught with other questions and challenges. But this center you mentioned that we just had the exciting news was funded just last month. It's not DARPA, actually. It's a related institute called ARPA. You take out the D.
Oh,
so
okay.
It turns out DARPA, it stands for Defense Advanced Research Projects Agency. That itself evolved. The D was added many years ago. It started from ARPA, Advanced Research Projects Agency, which has now been reinvigorated for health projects. So they've taken off they've taken the D back off. But other than that small correction, it is exactly in the spirit of DARPA. It's supposed to be fast, goal-driven projects that are contracts as opposed to more open-ended research, proposals and everything. And so last year, ARPA announced that they were going to really take a crack at what can AI do at managing all this information, just as you described, in the context of cancer clinics. So let's not worry about other diseases yet, but certainly you can imagine if you could make this work for cancer, and you could solve this information bottleneck, maybe then you could apply it to other diseases. This program is called ADAPT, the ARPA ADAPT program, and so my group is now and I should say my group is actually many investigators led by UC San Diego. And then there are 4 other such groups that have also been announced nationwide. We will create ourselves a network of AI centers for cancer therapy.
Wow. Yeah.
And so it's a very exciting prospect. So you can imagine it like this, Gill. Right now, when you are unlucky enough to have a cancer diagnosis, typically the first moves in this chess game against cancer are already prescribed. You get the standard of care first-line therapy. What, of course, gets more difficult is if this first-line therapy fails, and then second line fails, and so on. And worse yet, maybe your disease metastasizes.
And then you end up in immunotherapy. I mean, in the standard process.
Actually, immunotherapy is one possible action. That's one move you can make. There are other moves you can make, and these are not necessarily mutually exclusive moves. Maybe you combine a couple of moves either simultaneously or in sequence. Okay, and so currently these types of decisions are made in cancer clinics by something called the molecular tumor board. And you can think about these tumor boards as 12 human Jedi that sit around a council table and look at each case and try to make recommendations. As you just alluded to, though, the problem, of course, is the literature is happening so fast that these meetings become very anecdotal. Someone will say, "Oh, well, this patient has a mutation just upstream of AKT, which is a famous cancer protein. So therefore, I think they should respond to AKT inhibitors." Or someone else will say, "I just read a paper in the New England Journal of Medicine last night." That's how these discussions actually go. And sometimes they do arrive at good outcomes, but one sits and listens to these conversations, and everyone, no one's defending this, that is the best way. Everyone realizes something has to change. So the thought of this group is, can we, as a 13th Jedi on that council, put in place an AI system? And I think it's very important to describe it this way, because people will say, "Wait, you're turning your cancer care over to an AI. How do you trust the system?"
That's the only way to do it right now. When the information is dynamic and information is very high volume, no practitioner can keep up with it. It's an impossible task for a human. My only contention, though, Trey, that I wouldn't call it artificial intelligence. I would call it automation. It is really information automation to sort of extract the best information possible from a large data set that humans won't be able to do.
Absolutely.
So more practically, so how do you see, I don't know if this is in your research, but how do you see maybe 5 years from now? So patient walks into the clinic, there is a blood test done, maybe a bunch of other tests done, and the physician can see all those results. Maybe the machine, I'm just making this up, I haven't really thought about it. So machine analyzes all that data and gives some probabilities of what the situation might be. So then the human's role is to look at, the one with 75% and the one with 65% and say, well, the 75% one doesn't really make sense to me based on my experience. So I'm going to go with the 65%. Do you think we are heading in that direction?
I think that's exactly right. And so you have a table of possible therapies. You have a and then you have a probability, say, for each. And of course, you might also have recommendations that are off the book. You might actually, you can imagine value, certainly if you're in a drug company, which is the whole lifeblood of the drug company, is the novel therapeutics. But even in a clinical setting, you might imagine value in novel drug combinations that have not been tried or not have been considered, but are involving FDA-approved medications. So that's exactly right. And then in terms of the interface, I think probably large language models have given us the ability there you will just simply talk to this thing as a 13th member of the council. And importantly, these cannot be simply probabilities in a table and that's it, without any ability to interpret what caused, and I won't call it an AI, fine, fair enough, but what caused the system, what caused the system to make that recommendation or to assign that probability? You know, I think drug A has a 75% probability of success. Of a durable response, and I think drug B is going to kill the patient tomorrow. You cannot say that without, just as a human would never be allowed to say that on the panel without, well, why do you think that? You always must be prepared to answer the follow-up question. Why do you think that? And so this gets us into a domain called interpretable or sometimes explainable artificial intelligence. So these are not simply black box systems behind green curtains that aren't held accountable. They are, as we said, they're just the 13th member of a panel made up largely of human clinicians and scientists. And they will have a chance to opine on their opinion. Now, granted, they have read all of the literature. You have not. Right, they have read essentially everything. If you talk to the major purveyors of current large language models, even those, if the article has been published in an open way, not behind a paywall, the LLM has read it. You can basically count. So anyway, you know, you want these AIs to be able to get, or these systems to engage in discussions. And this gets us into a related topic, Gill. It's a lot of is generating a lot of buzz these days, called the AI co-scientist.
Yes.
To what degree can humans and AIs or machines work together? Let's just call them machines. Humans and machines work together to carry out the scientific process itself. And this is relevant on that tumor board. It's relevant in the clinic. These are physician scientists. You know, I mean, everyone who's ever watched House MD knows that for the most difficult patients, you sure hope there are teams of people behind the scenes talking scientifically about what to do for your cancer. You know, so I think there's a big open question: to what degree can modern machine learning work together with humans in some kind of collaborative scientific process?
Yeah. So let me ask you a loaded question, Trey. This is not your research, but I want to get your perspective on this. In a country with without a universal healthcare system, unlike the Scandinavia or New Zealand or something like that, we deploy AI, and the AI tells you this patient has very low probability of getting over it. And if the insurance company is swooping into that data, and increasingly all data is pretty available for anybody, then there are sort of policy and regulatory questions here, right? So this is a part that, I mean, US is a very complex system. We don't have universal healthcare. And we are increasingly going to make it with the big, beautiful bill that just came out. A lot more people without healthcare. This has implications, I think, from a policy perspective. What do you think?
Absolutely, and you know, I wouldn't dream of claiming expertise in any of those policy questions. All I can do is agree with your concerns and you know, say, look, there's a huge opportunity here. I think we know we need these systems to manage the information, and maybe well beyond simply managing information, generation of novel hypotheses and directions for care. But you know, wouldn't it be a shame if these systems developed wholly for good, you know, and for curing disease, were somehow used by insurance companies or other bad actors who would deny care? Clearly, that's the opposite of the intent at the moment, and so I fully support the need for studies and ultimately legislation here. But again, I wouldn't want, you know, I certainly am happy to defend expertise in AI and cancer at the intersection of AI and cancer, but I wouldn't want anyone to think that I have any particular clout in the ethical or moral or political issues that are surrounding it, other than to admit we absolutely have them, and we need lots of minds thinking about this, and we need engagement from politicians.
Yeah. Yeah, I mean, it's such an exciting area, Trey. I have done some work in this area for 3 decades. Presumably in the future, we could have some longitudinal aspect to it, right? The baby is born, we get some data on the baby, and we could have an AI system that sort of guides the baby's health over time. Some of it could be modifiable, some of it may not be. So as long as the healthcare is covered, we could progress toward an optimum scenario in healthcare, right? That's my hope, that humans will all pull together and get a more optimum point.
Well, and what you're talking about is not just curing disease, which is really where I'm focused at the moment. How close can we come to curing cancer, as a disease? You're getting beyond that into or ahead of that, really. How do we do prevention? And that's certainly an area where you and a lot of other folks have keen interest. And in fact, we've talked already, I think, on this podcast a bit about multi-cancer detection, ahead of people who actually have cancer. And I think your vision you've just painted of monitoring the health of the individual from infancy on is an extension beyond cancer detection. Obviously, it's the detection of all disease, but even more lifestyle recommendations.
Yeah, lifestyle. So I know that I have to let you go. So diabetes and hypertension, as you know, is probably half of our 4.5 trillion healthcare cost. And these are actually modifiable diseases if you identify early and prevent it by lifestyle or dietary choices or whatever the case may be. And so society could get a lot better than we have been doing. Prevention, I believe prevention is where the key is. For cancer, there's some hardware issues that we can't do anything about, as we have talked about. But there is still sort of behavioral and software issues that we could modify, right?
I completely agree. I don't know how you view this currently. How many, whether you're using apps to track your lifestyle and health. I actually keep those things to a minimum because I have the complementary concern here that I just can't live life, like a character in Brave New World, where you're constantly told what not to eat or and so on. So I try to live in a more loosely, by knowing generally how to behave, and when I'm not behaving, I feel guilty. But I generally I try to keep it a little looser, just so I don't. I'm not yet ready to train myself to my phone. For someone who, speaking for myself, can't even really bring myself to use social media properly, I think that's turned out to be a good decision in the long run. I get a little concerned about having these things control my life, but I do agree. I mean, it's very clear right now that what are some high level things one can do, and this gets also into what's well established and what's bleeding edge. What's likely to still be true in 10 years and what may change next year. And I think this absolutely applies to prevention and anti-aging and longevity. As a field, there's sort of very well established truism from the aging field and the prevention field, and that is eat less. Okay, it's
exactly. So I've been told that if you don't eat anything, you live for like 150 years.
Correct. Well, yeah, eat drastically less. Caloric restriction. It is well known in every species, essentially, in which one has ever looked, and the studies have been done over decades. That if you take a severely caloric restricted diet, or a moderately caloric restricted diet, you will live longer, right?
Yeah.
That's likely to be true in 10 years. Some of the the news articles you may read in your favorite journal or newspaper tomorrow about how to prevent aging may or may not be true in 10 years. Let us just put it that way.
Yes. Excellent, Trey. Yeah, thanks so much for spending time with me. And thanks again for coming back on the program.
Gill, thanks so much for inviting me back. Like the first time around, it was a pleasure. I'm happy to, as always, I'm happy to be here. Just give me the word.
Excellent. Yeah, thanks again. Yeah, exciting research. So I hope you keep pushing on it with a new project. That
Yeah.
seems really fascinating.
Well, yeah, check back in on the cancer oncology, the precision oncology.
Yeah.
It's just getting off the ground. We're going to have 3 years here to really produce some results.
Excellent. Thank you. Thanks, Trey.
Bye.